And welcome back. We're going to do our panel discussion now. I just wanted to make sure that everybody understood that in addition to the approximately 1,500 people in the room here, we have had as many as 90 people on the webcast. So, I think that's pretty good attendance for this. So what I want to do right now, and I'm going to go over there and sit down just so that I can make it appear to be dynamic for the cameras on the webcast, is just open up the floor for questions for the panelists, since we didn't have time for that yesterday afternoon. And then what I want to do is have a free-ranging discussion for the next hour about issues that were brought up in the various talks around anything from quality of data set to issues around integration, issues around sharing, issues around data management and manageability, and some of the thoughts to consider, like, what should be the focal point if one were to have a very controlled system. So I'm just going to leave it at that, and at this point invite people to ask questions of any of the individuals from the talks that you heard yesterday. And while people are going up, I'll just start. So in terms of the health outcome kinds of data, I mean, Brian, you said yesterday that the electronic health record system was never going to be perfect for a lot of reasons that I won't reiterate. Are there regions or countries where things might be better? Such countries that might have a single payer health system, for example, where the records might be more accurate and less open to bias, and should we be thinking about doing our experiments there?
Good morning, everyone. Can you hear me? Okay, so there's like two parts to that question, right? One has to do with the accuracy of electronic health record data, and one has to do with more administrative aspects, how widespread is the use of these data, how representative are they, et cetera. So that one's easier. There are some Scandinavian countries, Iceland as well, where, now I'm not an expert on this, but I do believe they have sort of some nationwide data collection. We don't have that, except we heard a bit about it from Francesca yesterday about Medicare data. So, but that's nationwide for a certain age cutoff. Now, in terms of the accuracy of electronic health record data, I don't think that should differ depending on the country. It's how physicians practice, and it's how they, it's how they use codes. And now maybe in places where payment doesn't matter as much, the coding is more accurate because it's only for one purpose. But in this country, coding is used for a bunch of different purposes. And so I think that, in this country, we're always going to have that difficulty. But I want to emphasize that longitudinal electronic health record data have so much richness that you don't have to rely on codes all by themselves. And so I think electronic health record data are fantastic for environmental epidemiology research.
So, I know that my HMO, or whatever I'm in, tries to put a real emphasis on prevention, and so there are a lot of statistics collected on individuals on an annual basis, at least. I mean, that's how we get our $100 back, is putting in our numbers. Is there any way to take advantage of that and link it with exposures from these kinds of records? Francesca, you can jump in too if you.
So as you were asking the question, and as Brian was commenting, I was thinking, so first of all, to address your question, well, you can always link exposure data and health data. The question is, in some way, the question is how accurate is going to be that linkage. And I think that, unfortunately, one of the, I would say, of the barrier, that's my own view, is that even though the health data either coming from the electronic medical record or from claims data, from Medicare data, or even if you design a pretty accurate prospective study, you can collect a lot of very good health data. The problem is that when you try not to link to the exposure data, often the exposure data, it's very crude. And especially for if you try to do a study with a very large number of subjects, you never really have the type of exposure data that you wish to have. And so if I think about in terms of the area of environmental epidemiology, I mean, I'm making a pretty generic statement, but most of the time, the major threat of validity or the biggest criticism, it doesn't come from the fact that you don't have enough health information or you were not able to link health to exposure. Is the fact that the exposure information is not as accurate as you would like. And even in the context of air pollution, which I think it's one of these environmental stressors where we measure, I mean, by citing EPA, it's really, they're doing, I think, a pretty advanced job. We have several thousand monitoring stations in the country measuring pollution. Even that, you never really know what's the individual exposure to pollution. So I think that we can link the data. I think there is a lot of information on the health data which we can take advantage. But I do believe that we have a long way to go to try to collect better exposure data. Of course, I am now referring more specifically to air pollution, but I think water pollution, it's even cruder, even less precise. And I think also for the other environmental stressor as well.
Let me take a couple of questions from the microphones. Lou?
Lou Gross, National Institute for Mathematical and Biological Synthesis. So this is a general question for all of you about the issue of time and data sets. So Francesca specifically mentioned issues of resolution and scale associated with integrating different data sets. There's also the issue, though, of quality of data changing through time associated with new instrumentation and or different methodologies or different survey methods and so on. And I'm wondering if there is a general need for a discussion in the environmental health community about how to manage time associated with data from a variety of different backgrounds. So it may be too big a question, but I'm just thought I'd bring it up.
Well, I can speak for the EPA. I mean, I was at the EPA on Monday, and I mean, you're exactly right. It's a huge, huge challenge, for example, with ambient level of air pollution. They're made available through the EPA. I mean, in one hand, I think really kudos to the government agency that collect the data and make that data available. But what's happened is that the data keep changing over time. And that is a huge issue for comparability across epidemiological studies. I mean, even among my own colleagues and my own team, we were dealing with this like yesterday. If you have downloaded air pollution data 3 months ago and you pollution data yesterday, you might have some differences. So, but I think that there is nothing that's beyond my control. And so I think it's really now getting coordinated with the EPA and say, well, can you give us, and there have been discussion after discussion about this, can you give us a freeze data set that even if it's not perfect, I feel even if a data set in terms of ambient level of air pollution, even if it's not 100% accurate, I think it's better if everybody uses the same data than have everybody using different cut of the data, because then when you try to replicate studies across group, you never know what is the difference. So I agree with you, it's a huge issue. As an investigator, as a scientist, that's something I feel I'm completely empowered to do anything about.
I wanted to, go ahead, let me, since the thought is on my top of my head, and then I'll let you talk, Elaine. I think the larger issue here is one of, as we collect complicated data and integrate across various data sets, there's a real danger of compounding the variability that's inherent in each of the data sets. And I think that has always been, an issue in any experiment, but the larger the experiments get, the more complicated, and particularly when we start to try and merge across data sets, we run the risk of either seeing nothing because of the variability or seeing artifacts because of the variability. I think the only rational way around that is to have really good characterization of obviously you want to minimize that, but variability is inherent in science. And I think that the issue that we have to confront right now is how do we quantitate it, and how do we track it over time. And, physicists always laugh at our biological experiments, and, when they realize how much variability that we live with simply from biological systems. But, it's time for us, I think, to, take heed of what they say in terms of, managing variability. And we're never going to get to the point where we can manage variability like a physicist or an analytical chemist does. The data is too messy. But if we don't understand it, and if we don't know where the sources come from, our experiments aren't going to be very meaningful. Elaine?
So those are extremely good points. And so I'll just follow up real quickly and say that capturing and characterizing that variability, especially when we're talking about exposure information, where I don't know, everybody feels like their variability is bigger than anybody else's, but we have little data but very big variability. And so, but what I was going to kind of go down a little bit different track, which doesn't answer your question at all, but just to throw in sort of this additional little twist in terms of time and data. So when you're looking at exposure, you're really dependent on human behavior and product use and diet and things that those not only is our ability to collect data accelerated, the rate at which our lifestyles and environmental stressors are changing has accelerated or seems to be accelerating. So finding mechanisms for even having the data to be able to say something about, so how are those changes in exposure and how are today's exposures impacting health is a big challenge. So it kind of goes beyond just the data itself to the metrics that we need.
And George, let me just comment that I think about biology very different from physics, in that in biology there is a lot of information in what physicists would call the noise, and that information is really important. So it's my attitude that we really need to pay attention to the variability.
Can I make the
variability is what makes things interesting, but yeah, it's different than noise.
Right.
Okay.
Can I just stay with that one point? Sorry, and maybe exactly what you were going to say. And this has to do with lessons from the physicists, and it has to do more with attitude, and it has to do with the fact that many scientists think of papers as the end state of their knowledge. And I think we need to shift to having more responsibility for assuming that people will want to work with your data and you need to work with others' data. So the aspect of variability that's in biology, I know that. But I think there's a more important point, which as funders, as publishers, as colleagues, I don't think we demand enough to know what was the context, what was all the annotations that take a little too much time to put down. And I think in biology that's really important. And I'm sure the same thing has to do with air pollution. And so I think we're getting these massive floods of big data. And what we're going to look back and go is, God, we had wasted decades where you cannot go back and add in the layers of information if the person who was collecting the information didn't have a sense of responsibility for how it was going to be used later.
Second, just to say I couldn't agree more.
A couple of very practical points, I guess. I really agree with this point about when you're trying to compare across modeling exercises, it's really important to have some sort of version that is the same across these studies. And for GIS studies and such, I wonder, I'm not familiar enough with them, whether they might consider having a quarterly version or a yearly annual version, and then keep a dynamic version as well. We're struggling with this from toxicology data as well, in terms of we're continually trying to improve our curve fits and our hit calls and our analysis of the assays, but we try to put out data in the public domain. I think it's really important then to annotate and to version that data so that people can use it and have some comparable basis for those data. And there's another issue that speaks to this with toxicology, is we have older data, historical data that will never be replicated. Many of these animal studies will never be replicated. The protocols used to generate those data have changed over time, and the challenge of trying to merge these data, bringing in these, it goes to all levels, I think, of data in terms of trying to decide how to deal with it.
So I'm going to do something incredibly dangerous, John Ballas with NIEHS. I wasn't here yesterday, so of course it's foolhardy to get up and ask a question. But along these lines, I think what I wanted to ask fits right in at this point. One of the projects I'm involved in is something called a metadata access tool for climate and health, where we're looking at a whole lot of different data sets, and we are providing a portal to the metadata. So if you're going to do data integration on a large scale, especially with data sets with which you're not intimately familiar, the way you figure out if you can and how you do it is by looking at the metadata. I was looking at family pictures, and one of my cousins said, you have to write on the back of it who is in there, because 10 years you'll go back and you won't know who's there. And that's exactly what the metadata are. They're the annotations that actually describe the parameters, the variability, the shortcomings, the context that we're talking about. So I have two questions. I don't know if this was raised yesterday, but the first is, if the panelists could first comment on whether, and so in the big data movement, there's a lot of efforts to standardize metadata, and so that the metadata are consistent, comparable, and actually useful for piecing things together. So some comments on how well we're doing with that, especially across disciplines. Are we doing as well in metadata for biology as we are for exposure and ecosystems, for example? And then the second question is, any great examples? Because I think we're still in very much a learning stage about how to really do good metadata standardization.
You mentioned the climate change and health, and I'm pleased because that's what I'm doing as well. And I don't know if that's addressed your question, but I'm going to be very provocative and direct about this. So, NIEHS put together this fantastic funding mechanism for climate change and health. And as you know, it's ending now. It was a series of R21. And I've been working on integrating databases that does a projection of climate in the future with historical data, with claims data. But with that funding mechanism, although I think it will be extremely valuable to do a metadata, there is no way I will ever be able to do it. So now you're saying that if your agency is doing something like that, it's interesting because I haven't seen, so I would love to learn more. But unfortunately, I'm going to raise the issue is that the R21 mechanism of 3 calls, which was asking specifically to look at the impact on health for climate change, required to link climate data to health data. And I think that all of that collection of R21 and many people that got funded, I think they have created some really interesting data infrastructure. But it's really too bad now, if you think about now that the call has ended, there has been really no mechanism to bring that data together and create the metadata. So. Of course, that's
Topic
my
for more
campaign.
discussion. But
Yeah, we should talk more.
We can
Yeah.
generalize it, though, for environment.
Yeah.
Hi, John.
Brian.
I did speak a little bit about this yesterday, but so concerning the electronic health record data that I talked about, there are about 20 members of the HMO Research Network. In general, these large healthcare providers have their data collected with software programs called Epic. They generally export data to a virtual data warehouse for analysis into studies. They are aware of the need for metadata. They are working to standardize that virtual data warehouse across all 20 sites to facilitate the use of data from 20 million people, which the HMORN represents. Currently, it's an underfunded effort, but they are aware of it, and if it was done, it would really enhance the ability to use data across sites, because right now, as you might expect, any laboratory test you might think of, there's huge between-site variation on numbers and on ranges and on prevalence of use, and it's not clear why that is. So metadata would make it very clear that that was a fasting, that was a random. We don't do it as often as that site because of the following principles, et cetera. So, so in terms of the electronic health record data, a lot of awareness of this issue, underfunded to date, not happening quite as well as it could be.
So one additional point on that is that, even though, say, for clinical chemistry evaluations in a clinical setting, you might have differences in baseline and variability and all that between sites, there are still minimum standards that are in place for those to be acceptable. And I think we have minimum standards in place for microarray experiments, for example. There are other areas, though, where the big data sets that we're interested in are collected without any, essentially even the most minimal standards of reporting or quality control. And I think that might be an area where it might be surprisingly easy but very useful to reapply some of the standards that we have in well-regulated areas, so that we can ensure that not necessarily variability, but we can ensure a level of quality and maybe just even recognition of what minimum standards ought to be from here on out.
And again, I want to make sure we're distinguishing the minimum standards for data and data quality and data variability versus the minimum standards and harmonization of metadata for the integration of multiple large data sets. So two different things.
Yeah, I do understand that, but I think one is dependent on the other.
Yes.
Pat Rizzuto, reporter with BNA, and I'm going to be more foolhardy I wasn't here yesterday, and I'm going to ask two questions. One, I'm wondering if there's a connection between this effort here on integrating environmental health data and the effort some agencies are making, like EPA and NIEHS, to do their systematic reviews of environmental health data for environmental health assessments. So that's one question. And if so, if you could help me understand the connection, and if not, why my mind shouldn't even be thinking there is one. And the second question is, what role librarians have in helping a scientist integrate the information for whatever project they have, and if you need librarians with special talents.
Actually, do you mind if I speak to the second point? Actually, it's something that we've been thinking a lot about at NIEHS. What's the role of the librarian in the library these days? I mean, we have this large space with lots of journals, leather-bound journals, and the reality is that most scientists and non-scientists who want to get access to the scientific literature use electronic resources. I mean, the NIH has a very robust means to get access to anything that you need on the internet. So we've been asking the question, what is the role of the library? And something I've become aware of is this, it's a new functionality within library science. It's called an informationist. And these are specialized, they're not librarians, but they're specialized they're individuals with specialized training. So they're subject area experts, but they also understand all of the details around library science and data management. And so you're beginning to see the emergence of this new informationist program. It's really taking hold at the national NLM, National Library of Medicine, and it's something that we're looking at very carefully at NIEHS. So does that actually answer your question? Okay. Actually, well, maybe I can remember a little bit. So you were, I think, referring to maybe some of the programs that are happening at the NTP, so what we have now are programs where there are scientists who are now reading the literature, looking at the literature, and coming up with reports. I mean, what's in the literature that we ought to be aware of? And so the extent to which this has been deployed, I think for the most part, people are looking at the published literature. But I don't know if Alan Deary is here, but he's actually specifically involved in organizing our databases so that our informationists can get access to this information so that the reports ultimately will be much richer. They will not be just a reflection of what's been published in the literature, but also a reflection of the vast amount of valuable data that's available in electronic electronic databases. I mean, one of our challenges is to figure out, well, what are the electronic databases that are out there? Which of them are good? Which of them have good metadata? And how do we use all of this information in a much more intelligent and productive way? Does that answer your question?
You weren't here yesterday, but we had some very stimulating, provocative talks by Steve Friend and Titus. And my experience is the library is good at sort of 10-year-old technology, maybe 5-year-old technology, at least in terms of accessing what's out there. But I think there is a layer that we're missing. It's sort of this very forward-thinking, how do we move our data into some of these really new capabilities on the cloud and in collaborative efforts and so forth. And having some expertise within these agencies to help facilitate. We've got little pockets of expertise within EPA that don't impact other parts of the agency in this regard. And I don't know how to bring that level of capability and innovation to the library, but I think it could really potentially be helpful in, pointing out sort of training courses that investigators who are generating data and maybe don't know how to really get it out there, the librarian could potentially connect them with people like Titus or people who are really doing innovative things in terms of data sharing. So I don't think we do a great job of it.
If I could just speak to that briefly again. So Alan is now in charge of a new office we're calling the Office for Information Science Management. And it's about going beyond the library. I mean, the library still has some functionality, but getting access to this information and planning your information needs at an institute is something we want to do. In fact, we're very interested in actually walking across the street and talking more with you, because I think it's actually irresponsible that we have our information repositories, you have your information repositories, and you have your ToxCast programs, and we have our Tox 21 programs. There needs to be a much more robust interface on how we're collecting data, how do we archive the data, how do we curate the data, how do we quality control the data, and then get it out there as a powerful tool for scientists to use. And we need to have the training programs so you don't need an army of PhD bioinformatics scientists to use the data. There should be client interfaces where the common scientist, the biologist, can actually use these data resources.
Hi, I'm Greg Farber from the National Institute of Mental Health. I have probably a naive question. What I'm really interested in, since we have a database that has information about human subjects, is how to link what we have to many of the databases that you all talked about yesterday. And what I'm wondering is there a reasonably simple link that we need to have in order to go out to what you all are working on? For example, if we provided for each subject what their age was and what their zip code was when they were that age, is that enough to get us linkage to the exposure data and the other things that we need? So I'm wondering, is there a common linkage like that that's reasonably straightforward? And if there is, I think a great outcome of this meeting would be to publicize that, to make sure that those of us who have human subjects data are collecting what's needed to make the linkage to the environmental exposure sorts of data. So I'm not sure who that's a question for, but I'm happy for anyone to take that on.
And briefly, I mean, there is a tremendous amount of interest and research. I can speak in the context of effects of air pollution on mental health and neurological disorder. It's something that I personally have not been very much involved on. But one thought is, and so I don't know to what degree in the community they're doing this type of research, they are aware of the existence of your database. But a very simple and maybe concrete suggestion will be that every time there is an RFA that is calling for research, for example, on health effects of environmental contaminant on the type of outcome that is collected on the database, especially if that database has been collected by the agency, to make it clear in the RFA and to say that there is a potential for the investigator to interact, let's say, with you to figure it out, way of linking the data. Specifically in air pollution, I mean, the ambient level of air pollution are by monitors. So if you know the zip code of residents, potentially you can link them. So I don't think that there is a major barrier. I'm wondering to what degree it's very well known in the community, and maybe that would be an additional piece of information to put specifically in the RFA, knowing also that there is an opportunity to interact with you, to think more creatively about different way of linking the data.
Oh, no, you can't. I guess I'm going to go back to this exposure data issue. So things like air pollution, ambient air pollution data are very publicly accessible. And so, at one point in time, I was trying I was trying in my own little way to get, for the EPA STAR grants that I was involved with, to try to get exposure core requirements for exposure cores into those kinds of grants. Because part of the issue clearly is that the investigators doing the health studies just simply don't have expertise on the exposure side, and if they were just required, since they don't seem to have those connections on their own, if they were simply required to have that expertise in their grants, applications and teams, that there might right there and then be that potential. Now, in general, the air pollution and ambient air pollution data is an example of the very best exposure information that's really available. This is not universally available. It's not available on any indoor environment, and how much indoor environment and diet and other kinds of not more, but equally direct and relevant exposures. There's simply not that kind of general geographically wide, population-wide kinds of information. So linking it is to something that's not there is definitely challenging. And then just the other thing that I would say in terms of human studies, that I find to be the most challenging is, while the people doing these health studies can access the limited high-quality exposure information that's out there, some of the power that I think we're losing is that these individual health studies, because of so many human studies and confidentiality issues, can't link. We can't do the read across and linking and hypothesis development that we might be able to do, should we be able to address some of those issues. And I know NIEHS did have a workshop, now I'm already forgetting, it was within this last year, that was very focused on bringing their grantees together and a range of other people to start talking about this. But it's a huge problem, and one we can hopefully figure. I guess this is my thing about So which pieces of those studies could you make accessible without, and it's not going to be at the resolution, and it's not going to necessarily allow individual associations, associations at the individual level, but would there be power anyway across studies in making some pieces and some aggregated data accessible and publicly accessible to, sort of start that discovery process so that then you could dive down within studies?
Yeah, I just had a quick question specific to your database. So NIEHS had a workshop about two years ago on autism, and we attended that in the thinking with our database, and then went back and actually curated as much data as we could that was associating autism with chemicals or genes that have been associated with autism and chemicals, and so. But that's not our area of expertise, and so I think, again, it's there's a need for a mechanism to sort of bring these groups together, and I'm virtually certain there are ways that things could be connected. It's just a matter of sitting down with data models, looking at where the overlap is, and making those connections. And, of course, that can be done at a range of levels, simple links to actually incorporating data into our various databases. So I think there's a lot of potential there that's untapped.
Brian?
So Gregory, yesterday we heard a couple of examples of successful linkage of different kinds of data streams. And both of those, Francesca's air pollution and health, my various studies, were both characterized by the ability to characterize exposure, using that term generically, in space and time. And so with what Ann talked about, with what Carolyn talked about, those data sets do not allow you to characterize things within space and time. Now, when we characterize in space, it's at the exact latitude and longitude. You suggested asked whether zip code was good enough. Well, Francesca did aggregate various data to the zip code level, and I think that can be successful with regional scale air pollutants because you can capture the necessary variability, but for not for all exposures will a zip code centroid be adequate for this kind of stuff. The big challenge is, so in general, we've talked yesterday about the two examples that were successful were things that generally got to people through the air. I don't know how my MRSA risk travels from these animal feeding operations, but it could be through the air. But the bigger question is, what do we do with stuff that gets to people through diet or through water? And you actually have to measure or estimate an individual internal dose to discern health effects. I heard nothing yesterday that gave me any ideas how to use these various data sets to do that. And I would love to think more critically about that today. How can we make the 5 things that we talked about talk to each other? That's, to me, the big question.
Some questions from...
I've got a question about the electronic medical records. We at NIEHS have been thinking a lot about how do we get more environmental exposure data into the electronic medical record. But a question for you is, does it really belong there? Or should there be, say, a unique identifier for a patient which cross-links to other databases that relate to that individual's exposures? So, I mean, for example, we talk about putting the whole genome sequence into the electronic medical record, the exposure data into electronic medical record. Is that, again, where does it belong there? Or should we be developing robust means to be connecting different data sources, so that in a single query, you can seamlessly query multiple databases and get decentralized information coming together? I mean, what are your thoughts on that?
I think it's a great question. I'll say something maybe provocative, but I also see patients, and I see patients who believe they have environmental occupational diseases, and often, in my specialty and in others in the room, the cause of the illness matters. If it's an environmental or an occupational cause, then there could be implications for other people in the home, other people at the work site, et cetera. So we spend a lot of time trying to figure out where the disease came from. But most clinicians who are treating disease don't really care what the cause was, whether your lung cancer was caused by bischloromethyl ether or by cigarettes or by or asbestos. They don't really care what the cause was. They want to get you into the oncologist and get your surgery and get your treatment. And most treatments don't change by cause. So I wonder whether it's all that useful to the average clinician who's not a specialist in this field to know cause. And if it's not important, then you're not going to get health systems to put that information into their electronic health records. You know, we have the ability at all these sites to add new stuff. And many of you are aware of the American College of Occupational and Environmental Medicine has a proposal now to get a job title and I think an industry code into the electronic health record for every patient. That's nice. It's not very much, and it won't be terribly useful, but it's not a bad start. Whether we want to do more than that, I think you do have to convince the health system that it's important to medical care to have that information in there. Otherwise, they don't want to take up that space. So I think for the time being, we're going to have to think about how to do it other ways.
Titus, let me say one thing before you start, which is for the people on the webcast who have posed questions, they are being displayed such that the panelists can see them, but at the same time, we've got lots of questions in the room, so we'll get to them if we can. Thanks. Sir.
I'm not sure the panelists can see them. Oh, we're right up close. Okay, I was like, I don't.
We have a special secret thing.
So with regards to the metadata and also this other topic that we brought up yesterday of that I think Barbara brought up of open source and making the methods and so on available, you know, there's an issue with the incentives, as I think everybody in the room that talks about data has two hats, right? There's the database hat, which is it would be great if everybody deposited all of their polished metadata labeled data in my database so that we could make use of it. And there's the other hat, which is the person trying to publish primary research papers. And if when you try to publish primary research papers, it's hard enough to get the damn paper out. I mean, getting the paper out, getting it past reviews, getting it published, doing all the formatting, et cetera, is annoying. And if you have to add as much or more work to get the source code and the data out in a nice, usable form, then it's sort of an intolerable burden. And I've watched this in my lab where some of the people in my lab have a better time of it because they've been trained better, and others struggle a lot more. And so the burden then falls on me, which doesn't work out very well necessarily. So there's this question of, I think Stephen and I both want to talk about incentives all the time. And so there's this question of how do you incentivize this? And funding is definitely one way. But there's another set of social manipulations that you can indulge in. And so we saw, I think Greg mentioned yesterday in the X-ray crystallography community, if the tools that you use to analyze your data automatically make the data available in a form that can be the metadata and data and analysis results available in a form where they can be uploaded to the common database, that's great. But then there's other things where if you can make the data more useful for the producer of the data by when they add their metadata, then they'll do so. So one example in the field of metagenomics is there's a server called MGRAST. And MGRAST is basically, it will take your large sequence files and give you a set of analyses with bar charts and everything that you want. And they insist before they will run an analysis, you have to both upload your data, provide metadata, and accept that they are going to publish your data within some period of time. I don't remember if there's a sunset period or if it's when you publish it. There's a single button to make it available that provides an accession number for your publication, right? This solves all these problems associated with where do I put my data, how do I make it available for publication, and in order to get a useful analysis out of them, you have to annotate it with metadata. So that kind of approach has the benefit of encouraging people to do it without punishing them when they don't, and makes it easier to provide your metadata and more useful to provide your metadata than not, which is really what we're all looking for. And so there's lots of social tricks. Stephen talked about gamification yesterday, and I think this is sort of the reverse for data. We'll run our nifty algorithms on your data if you give us enough information to do so. You know, we'll correlate your data and metadata with other data sets based on the metadata annotations you provide, thereby leveraging the power of our database and our computation on your behalf. for this much extra work. And then there's a really neat trick that I didn't come up with. It comes, I probably shouldn't, I don't know who in the organization did it, so don't blame Paul. But so Paul Sternberg at Wormbase noted that if you post slightly wrong or slightly naive annotations in a genome database, the next day you will get 5 emails from 5 different professors correcting you. So it's a great way to get those annotations, in detail updated. There's all these little tricks that you can take advantage of the way academia works. I'm not recommending you post wrong data, but if you post shorthand data that it doesn't acknowledge the contributions of people, those people will step up and tell you about their contributions. So there's all these little tricks. And I think that is in many ways better than giving people a pile of money up front, or not giving them any money and demanding it, or giving them a pile of money up front and seeing if it works out on the other end. It also has a side benefit of making sure that the metadata that you do provide is useful, that the metadata that you demand is useful, because if it's not useful and you're not using it in any of your software, people will have no incentive to provide it, and you won't have any analyses that make use of it, which addresses a lot of my concerns about the ontology and metadata world development as well. It's not immediately useful to me. It's hard to get me to use it. And then one quick second comment. We've actually been funded on the question of data training. We just received an Office of Cyber Infrastructure supplement at the Beacon Center to run workshops on data and metadata training. And we're going to hold an organizational, I'm not sure what the right word is, an organizational meeting on that in probably 6 to 8 months. Drop me an email if you're interested in attending that. Drop me a separate email if you're interested in hosting such a workshop as a biology group running databases interested in, how to train people to do this properly. Because we don't really know how to train people to do it properly either. That's part of what the funding is about, figuring out.
Any responses? Mine is, I try to use those same tricks on my children, and I believe my wife probably uses them on me. So.
I just want to mention we're struggling with this within our program, our ToxCast, Tox 21 programs. Gentleman from NIHS mentioned NTP. We had a retreat the day before I came to this meeting, and the book by Nate Silver was brought up. We're trying to think about, and I think it's the third rail of what you just spoke about, Titus. It's for the data generators who don't necessarily have all the expertise to do the type of public projection of the data that we'd like to. How do we maximally leverage this data to get people to use it? We haven't been extremely successful at this point. We're trying to work on that, as Matt mentioned, with the dashboard effort and with other efforts. How do we get this data out? We've talked about ideas of like a prediction contest, not really contest, but putting data out from the standpoint of asking people to be critical, asking people to actually run their models. And you've actually planted the idea in my mind of as a price for this, or to try to encourage people to then upload something that enhances our public projection of the data so that it makes it better for everyone in the community, as opposed to just putting it out there and it goes in its 500 different directions. So maybe we can think about that as well as we move forward. But it's a challenge for us as well as data providers in a small group.
Just as a further comment of what Titus was saying, I think in this context of big data science, the pace of scientific discovery is tremendously accelerated, not by publishing the next paper, but by understanding how the data were being linked and analyzed to publish that paper. And I found it's very frustrating, I think, for everybody in this room, the fact that it's so hard to figure out how to do better, but at the same time, I think right now is the most important thing that we have to solve, because I think that the pace at which we're publishing paper is so fast that even ourselves, we have a hard time to track the next paper. And now, there is always that X paper, even if it's a high impact paper, and say that, exposure to X, increase the risk of Y. But that's not really what the science is behind. Right now, the most important piece of the science is how you got to that conclusion. And so I'm not saying anything new, but I'm just to say that this is really the most important thing that we have to figure out, and how it seems to me that it's going back to incentive and the role of the investigator. I think right now, impact, at least I can speak from my own institution, impact, the impact of your work, it's becoming extremely, extremely important, at the point that now measures like the H index, which I'm sure people have different opinion, is becoming a key factor in the promotion process. And so another incentive is that if you start making your data available, or actually not even making a data available, but making clear what has been the step that you have taken to publish the paper and how you have linked the different data, can tremendously increase the impact in your work, and that would be another measure of incentive. But this is really, I think, I mean, the type of discussion we're having today, I feel it's really one of the most critical pieces for scientific advancement right now.
So just one additional point, as you were talking, Anne, that I was thinking about is, you know, in the specific example around using chemical structure-based data sets for toxicology, you know, we've done a fair amount of evangelizing out there with various regulatory agencies on how to do that and how to use that. And one issue that often comes back is that it requires a different set of expertise and a broader set of expertise than they're necessarily used to dealing with. And so, you know, maybe one of the things that we can do is figure out ways, at least virtually, to bring together physical chemists, medicinal chemists, toxicologists, you know, kineticists, metabolism experts, all together in one place to help people get comfortable in using these things until such time as you know, they decide that they need to have that sort of expertise in their own shop. So, Bill?
Bill Farland, Colorado State. So I want to try to take us in a slightly different direction here. One of the questions was the challenges to trying to integrate these things, and particularly for environmental health. So I guess what I want to do is to let us think a little bit about the Matthew effect, or the iceberg effect, or the lamppost effect, however you want to characterize it, which is, in my mind, the focus that we have had on a very limited set of chemicals that we're interested in, and that we collect data in, and we try to take into epidemiologic studies, and then contrast that with what we heard from Anne about how carefully we're trying to look at the chemistry of a particular chemical that goes into a relatively simple test, with what we heard from Stephen, which is when we get into people, we have complex exposures, we focus on an individual chemical, and we've got a very, very wide range of toxicodynamic responses to that. And so how do we take this kind of a perspective and say it's actually feasible to start to integrate these kinds of databases?
I'm the wrong, I actually wanted to follow up. I'm trying to understand your question. So are you talking about the differences in scale in terms of the global nature of some of the data versus the very focused specific nature of other data? Or did I not?
So just as an example, I mean, we, let's take formaldehyde. We do very careful evaluations of the chemical that we're testing, the kinds of adducts that we're seeing. We do a lot of good work there. And then we decide that we're going to look for an epidemiologic study in formaldehyde. And we say, well, there were people who were exposed to formaldehyde in mobile homes. And we basically know that that was a very complex exposure. They probably were exposed to higher levels of formaldehyde, but that was still a very complex exposure. And we essentially attribute the responses that we see to formaldehyde.
Yeah, see, so that, I mean, that's one of the things that's been bothering me a lot about the exposure information and the advancement or lack of advancement on that side of the equation is that we are now able to do a lot of these global, more discovery kinds of, well, global collection on susceptibility, on expression, gene expression, and response, but we're still not doing global measurement and evaluation on the exposure side. So why do we believe, like it's crazy to believe that those differences in resolution and scale aren't impacting what we're looking for, how we're looking for it, and the conclusions that we're coming to. So I do feel like there's this lack of balance on these sides, and I do think that sort of these very recent discussions on what would you measure for something like the exposome that starts to more globally get at the exposure side are really important. And I'm seeing some questions and comments from people on the watching or listening on the web. And a lot of them and a lot of the issues that people raise when they're talking about integrating data and linking data go back down to if we don't connect the exposure to the dose, nothing, it's all meaningless. And if we don't do it at the individual level, it's all, we're not going to be able to answer this question or that question. And I think there are, different kinds of questions. And one of the speakers, at least one of the speakers yesterday, probably, I think maybe a couple, raised the issue that, there's different things we want to be doing with these data and these information, and these new global tools really provide this opportunity to get at maybe looking at what we don't know, and that's a really important, it's a very different approach and a very different question, but it has the potential, if we can do some of this linking on the global side, where we're not going to get these individual connections in the same way, but what we're going to do is potentially identify things we might want to investigate, and in fact, potentially we would start to investigate things that might be more important than where we're currently putting 20 years of formaldehyde research. I'm more than 20 years. See, I only started 20 years ago in formaldehyde.
Just a quick reminder, what I presented in ToxCast in particular, Tox 21 to some extent, was to address this question or this problem that we have literally thousands of chemicals out there historically predating EPA's regulatory authority, outside of EPA's regulatory authority, that are out there with no tox data, virtually no tox data, virtually no exposure data. And we can continue to try to, work on formaldehyde and chloroform and a few compounds that we know lots and lots about. There is the mindset that we should know exactly how to do all of this before we start on the next chemical, but I think we have to do both. We have to try to figure out what ToxCast is about. It's about prioritizing. There are efforts within our group, perhaps Matt can speak more to this, to try to estimate internal dose based on reverse toxicogenetics to see if the dose at which we're doing our high throughput screening has any relationship to a dose that might be actually experienced in the environment. So we're coming at it from both sides, and the many chemicals versus the very few are very different problems, I guess. I don't know precisely how to connect them, but we're trying to fill in that middle ground.
And one thing, Bill, the problem in epidemiology studies is the same as it's always been. And I think one of the frustrations for me as a toxicologist is how little the toxicology data gets used in ruling in or ruling out biological plausibility. I think one of the advantages of the databases and